medicinal plants. The products are locally prepared, and we work on a referral system, especially after Corona, COVID-19. Even the conventional doctors are referring patients to us, especially chronics, cancer patients, arthritis, respiratory system chronics. All these are being managed through herbal medicine. I welcome you to my session. It is not that much, but we are going to look into what is traditional medicine, the role of traditional herbal medicine in preserving cultural heritage and promoting health. That is community health in a Kenyan setup. I welcome you all, and my gratitude and thanksgiving acknowledgements go to our Nairobi University who have accommodated this event, and we thank, on behalf of the Herbalist Society of Kenya, I give thanks for the invitation that you have extended to us. When we talk about traditional medicine practice in Kenya, there's so much. There's a pillar of cultural heritage, that is, traditional medicine is an integral part of Kenya's cultural heritage, and for centuries, our communities have relied on the wisdom of herbalists and traditional healers to treat ailments and promote health. This knowledge is passed down through generations and is not merely about health, it is also a rich tapestry woven with the threads of culture, history, and identity. The current situation, analysis and insights of traditional medicine in Kenya. Knowledge preservation and transmission. Traditional knowledge is primarily passed down orally within communities. This mode of transition, while culturally significant, is vulnerable to loss due to modernization and the passing on of our older generations without proper documentation. Meaning, if we are not going to work hard and do documentation of the knowledge that the traditional healers have, very soon the modern generation will lose the real formulas, the real knowledge of the medicinal plants, which are also extinguishing. Integration with modern healthcare. While there have been efforts to integrate traditional medicine with modern healthcare, these efforts are often fragmented. There is still a lack of formal recognition and support for traditional healers within the mainstream healthcare system in Kenya. Research and validation. The scientific validation of traditional remedies is crucial so that we can turn that knowledge, that indigenous knowledge, on patient care, on identification of medicinal plants, on preparation of the herbal medicines. That one is lacking. There is still a lack of formal recognition and support of traditional healers within the national healthcare system. When we come to research and validation, research in this area is often unfunded. It lacks the necessary resources and institutional support. When it comes to students coming to a traditional healer, maybe the student who has come to gain the knowledge on traditional medicine is given support. But who is going to support this traditional healer? Who is going to go to the field and give all the knowledge to the students? Furthermore, after that research has been done, the information has been collected, but what happens? What is the product? What's the outcome? Is it just the papers in the shelves, or we have products that are there in the chemists, that this is a product from a certain university which worked together with a local traditional healer using the knowledge of the the traditional healer, and now there's a product. For example, during Corona, it is so surprising that even the people who used to talk about traditional medicine, they turned out to be herbalists. Everybody all over Kenya. They were taking ginger and the hot water. I really don't know, from the science part of it, I really don't know whether hot water and the ginger could kill the COVID-19 virus. But you find that in every home, everybody was talking about herbal medicine, traditional medicine. We used to hear there's a traditional healer at such and such a place. So they would go and look for some herb there. What we are talking about as a traditional healer, we mean after this, you students and the professors and the owners of this institution which is doing this wonderful work, what have you done to the Kenyan patient? Are you going to have the papers written, kept somewhere? Or you are going to come back with a fund and say, now, through Dr. Lydia Machoke, can you come with your traditional healers for a symposium like this one, so that we can be able to collect a few samples that you people, you traditional healers, Lydia included, you have been claiming that this one treats and cures some diseases, so that your research can be real, so that we can be able to empower our Minister of Health. Where there is failure to cure some diseases, the traditional medicine products should be there in the shelves of the chemists, so that the patients can do what? Can gain from the research. Because this investment, this is an enormous investment. For you people to come to Kenya, you spend all that which you have spent. What is the outcome? What answers are you going to give? Is it just a certificate after the students have graduated? I heard most of you are PhD students. And that's why in my introduction, I say, when I was doing my PhD, I made up my mind, I'm going to be a clinician. I'm going to deal with the patient. So that all those papers, my thesis was the traditional medicinal plants used by Abagusi, a tribe in Nyanza, where I come from. So I wanted to turn that thesis into a reality. Now what is remaining? It is you people, the chemistry person, to come back and say, this is the value we are going to add into your product. We can identify the medicinal plants. We can surrender our formulas. But who is going to hold the hand of the traditional healer and walk with them, so that we can solve the problems in health which have never been solved? Conservation of medicinal plants. It's okay that we have a lot of medicinal plants within our region in Kenya, but what have we done? Harvesting from time immemorial, it is world harvesting. So even if we came up with a product, are we going to sustain the supply? Meaning, we must first do the documentation of this knowledge on the medicinal plants, so that we can be able now to start documenting the knowledge on how best we can be able to start growing and doing conservation of the medicinal plants. So that if we come up, work together, we collaborate, we can be able to sustain the supply for eternity. All through, when we talk about Panadol, maybe the formula can change, but Panadol will be there. So if we talk about, let me give an example. We have Puriafu. Puriafu product is a product which we prepare from Prunus africana. How much Prunus africana have we grown in Kenya? So that if we have to have that product in the shelves, there will be continual supply of the raw materials. So the part of conservation is also a challenge, and that is what we need to look into. What is our vision? What is our future expectation as traditional healers? Documentation and digitizing traditional knowledge. That one I've mentioned before, it is paramount. It is paramount for research to succeed. So that you don't have to come to Kenya to get the Kenyan traditional healer. You will get that knowledge within the websites of the traditional healers. And that is why within our institution, that is the Herbalist Society of Kenya, we have the younger youth. It could be they don't know, they cannot identify the medicinal plants, but they are there, they are members of the society, so that they can be able to assist and help the traditional healers document and keep their records of the work they do and the knowledge that they have. Formal recognition and research, advocacy for formal recognition of traditional medicine within the national health system is crucial. This includes licensing of traditional healers, providing training, and integrating their practices with modern medical protocols. That one is missing because we don't have laws and regulations within the department or within the Ministry of Health to regulate this, what the traditional healers are doing. Then we have sustainable conservation, that one I've said something about it. The challenges we have right now with traditional healers, as younger generations move towards urbanization and modernization, the economic constraints, the regulatory and the policy barriers, we don't have a clear regulatory framework, even supportive policies. So the younger generation, they don't really understand what is this traditional medicine. And as they move forward, even you researchers, you young researchers, you will miss the know when you come to work in Kenya. You want to work with a traditional healer, you will not get to understand if we are going to miss the part of documentation, the part of regulations, and better systems to be put in place. Then we have a challenge with intellectual property issues. This is a big challenge which is facing the traditional healer in Kenya. The students come, that's okay. The researchers come, that's okay. The research institutions come and approach us. They want to give us the students. But what happens when you complete your thesis? When you have been given your certificate, you have your master's or you have your PhD. Have you recognized any of those traditional healers? Do you recognize them? That one is lacking. And I think that's the cause of why the traditional healers sometimes they refuse to disclose what they know and what they do. Because they have learned from time immemorial that we, the academia, we don't recognize them. We don't acknowledge them. Even just acknowledging them. That this knowledge, I have written this thesis, it is the knowledge of so and so. I worked with so and so. That one is also missing, and it is a challenge. So we need to work together and be able to move forward in a well systematic way so that we can be able to uplift the traditional healer. The action points, that's the way we are going to move forward. We should engage the community in education so that the community can get to understand what is traditional medicine and the benefit of alternative therapies, either within the hospital or within the clinics of the traditional healers. We should initiate this program and engage the younger generation about the value of traditional herbal medicine. Encourage the integration so that we can be able to get the knowledge transferred from the traditional healer to the mainstream healthcare workers, because they lack that knowledge of what we do. They are nurses, they are clinical officers, they are doctors, but there's lack between the traditional healer, the knowledge they own, and the conventional doctor, the knowledge they own and the way they work. Policy advocacy, we should work with policymakers to develop supportive frameworks that recognize and integrate traditional medicine into the national healthcare system, advocate for inclusion of traditional healers in the health policy discussions. Research and development, establish dedicated research centers and secure funding to study traditional remedies. Encourage collaboration between universities, research institutions, and the traditional healers. Conservation initiatives. Launch community-driven conservation projects to protect medicinal plants. Promote sustainable harvesting practices and habitat restoration. On that point, the Herbal Society of Kenya, through the initiative of Hope Natural Healthcare Center, we have been training the older folk, that is the aged traditional healers. What are we doing? We want them to be enabled to get to understand that once they disseminate their knowledge, that's the only way this knowledge can be preserved. Because once an old person has died with that knowledge, that is final. Then on the part of intellectual property protection, we should develop and implement policies to safeguard the intellectual property rights of traditional knowledge holders, ensure fair and equitable benefit sharing arrangements. In conclusion, preservation and the promotion of traditional herbal medicine is crucial as part of our heritage in Kenya and in Africa. So for the health of the nation and well-being of the communities, the gaps which we have mentioned there, they should be sealed by collaborating between the traditional healers, the ministry concerned, but the Ministry of Culture and the Health, the research institutions, and the real intellectual property who require the intellectual property to be preserved and safeguarded, that is the traditional healer. Thank you.
Thank you very much, Lydia. So any questions for Dr. Kimuto? Any questions?
Yeah, thank you for the wonderful presentation. I just want to comment on the issue of intellectual property you mentioned. I know intellectual property with the cultural herbal medicine is a global issue in many African countries. So just wondering whether as an association, Kenyan Medicinal, okay, thank you. As an association, maybe if your intellectual property will be protected by a law, so that you can be able to take legal action when your intellectual property is used, especially for commercial purposes. So I don't know whether that is in place or is something that you are working on. Thank you.
What we are doing right now, there before, the traditional healer was not included in any of the policy making processes. But right now, I'm part and parcel of the policy making on intellectual property rights, KEPS, Farms and Questions Board, so that at that level, the traditional healer can understand and know what is going on from the part of the government. But as of now, the laws and the standards and the research standards, it is now when we are working on them. It is now that the traditional healer has been included in those committees.
All right. Just one more question now, so that we can catch up with time.
Thank you so much for the wonderful presentation. I know in the Ministry of Health, we have a Department of Traditional Medicine. So I don't know whether you are doing any collaboration with the department.
That's exactly where I work from,
Okay.
and I was part and parcel of the committee which is working on the regulations. And we have a bill, a pending bill, which we want to take to Parliament, and that's why I mentioned about campaign. We have to see the policymakers, because if we don't work together with the policymakers, they will get this document and throw it down the table. So I'm part and parcel of the process.
Just one other question.
Thank you so much, Dr. Benito, for your presentation. I want you to help me understand whether in your clinic, how many people do you receive, maybe monthly, and how many people for real get healed? For real, for real.
For real. For real.
Yes.
Allow me to say something in 3 minutes. At the age of 27 years, I was diagnosed of breast cancer. I went through surgery. I went through chemo. I went through radiotherapy. This is the note which discharged me from the Neda hospital. Take her home. Whispered into my husband. The doctor forgot that you can be a cancer survivor who knows and understands English. So the doctor mentioned to my husband, take her home, not even to Nakuru, take her back to Nyamira because she doesn't have more days to live. That time, my grandmother, whom I inherited from, she was over 130 because she died in 147. She was 147. She died in 1992 at the age of 151. She called for me. She said, I will not die. She's my inheritor. I'm going to show her the roots that she's going to boil and take. The bugs that she's going to boil and take. And the traditional vegetables that she's going to live on. And the wild fruits that she's going to live on. This is me. My operation was on 14th of January 1988. Today, I've gone back to class. I was not a medic. I was an accountant. I'm a former KCP manager. I went back to health sector. I started from nursing because I wanted to care for a patient. After that, I had to go and do chemistry for my degree. After that, I had to do the study of medicinal plants, which my PC people have been using. I come back to Hope. In a day, at Hope Center, we have a laboratory. I don't just sit, or my other nurses, they don't just sit and listen to you as a patient. You got the whole system. We do a lot of record keeping. We take your name, your age, everything, your contact, where you come from. The complaints that you are presenting and what the laboratory person is going to tell us is wrong with you according to what the doctor is going to request from the lab. We keep that record. So when I talk about in a day, like the whole of April and May this year, I have 36 cancer patients. That is April and May. Meaning something is going wrong in Kenya. Why are the cancer patients going to traditional healers? What? Arthritis, in quotes, be it osteoarthritis, rheumatoid arthritis, and the like. Yesterday alone, between 8 o'clock and 11 o'clock when I was leaving Nakuru, I had treated 9 people in a day with arthritis. I have 17 sickle cell children. What can I talk about? Infertility. Infertility. How many men who suffered from mumps, they lack sperms, they don't get children. They are ever treated of UTI, and the lady is told she's the one who doesn't get children. And yet, there is medicine which we can give or which we give. Infertility is my first number one patients. Forget about cancer. So, Prof, it is just a matter of my traditional healers to continue keeping records of what we do, where the patient comes from, the complaints. We collaborate with the laboratories. I'm encouraging all the traditional healers in Kenya to collaborate with the modern laboratories. Although, me, I have a unique laboratory. I'm Chinese, my background is Chinese, so I have all the laboratories within my setup. So asking a traditional healer how many patients you see in a day, I treat my patients over phone. 3/4 of the people I meet in conferences, in symposiums, in wherever.
And they get healed for real.
I think I should be the first one. I think you should take some biopsy today, so that you can prove whether I'm cancer free or not. For that 7 years, I have enjoyed life.
I wanted her to really just to make sure that what she's telling us is real, because many of us, and I'm sure that many of us have got their own biases regarding traditional healing and healers. And so that's why I'm kind of saying real, because you have seen it. And maybe next time when we come next year, we'll bring some exhibits, people who have been healed. If they want to give a testimony, that would be wonderful. But you are a testimony here for us. Yes,
37
wonderful.
years.
37 years healed of cancer. A hand for her.
Let's say something about indigenous knowledge. Right now, prematures are put in machines, isn't it? In incubators. I was born 6.5 months. I'm a premature. My grandmother brought me up at the fireside. They used the fireside, that heat, that warmth, as an incubator. So either I'm a miracle or for real, indigenous knowledge works.
Another clap for walking miracle. So our next speaker is Dr. Irungu, so I'll give you 10 minutes for your talk. Then questions and project. Meanwhile, you can introduce yourself. Just to save time. Connect you. Don't worry about connecting.
Good morning. I'll be asked to introduce myself. My name is Jerry Irongo. I am a research scientist with Kenya Medical Research Institute, and I am alumni of the University of Nairobi. I studied chemistry and mathematics for my undergraduates at the University of Nairobi, and I did my PhD at the University of Nairobi with Professor Abi, who was here in the morning. He was my supervisor. And I work at Kenya Medical Research Institute, the Center for Traditional Medicine and Drug Research. And I've been asked to briefly come and say what is it that we do as a Center for Traditional Medicine. As our name sounds, a lot of our work is centered around traditional medicine, but we also do many other things. But for purposes of this presentation, I'll only focus on traditional medicine. And because traditional medicine is broad, we limit ourselves to herbal remedies. So when I talk of traditional medicine, it's limited to herbal remedies only. Yeah, and as he continues, I can say that Kenya Medical Research Institute, for our visitors, it is a government organization, and we are mandated to carry out research in human health. Yeah, I said, and then our vision is that we would like to be a leading center of excellence in human health research. Our focus is human health research. And our mission is that we would like to improve human health and quality of life, and we can do this through research, capacity building, innovation, and service delivery. And our motto is that we are always in search of better health. And briefly about KEMRI for our visitors is that we have about 14 research centers. They are not just within Nairobi, we are spread in different areas of Kenya. And the 14 research centers, they focus their research areas on any of the 7 programs. So like us, the Center for Traditional Medicine and Drug Research, we focus our work on natural product research and drug development. And briefly about the Center for Traditional Medicine and Drug Research, our mandate is to carry out research on herbal remedies. And by research, I mean we look at the safety, we look at the efficacy, but we also try to do drug development. But besides traditional medicine, there is also a lot of work on biochemical analysis and pharmaceutics research. And we have our scientists coming with varied different backgrounds, as you can see, from biologists, botanists, chemists, and the like. And our department is, we have 4 divisions. We have a botanical sciences division, chemical sciences, biological sciences, and pharmaceutical sciences. And I'll take you through what each department is mandated to do. Like, for example, our botanical sciences, they do documentation of medicinal plants. How do they do it? They rely on our research scientists. Research scientists carry out independent research activities, and whenever they do a paper or they do a proposal, they have some background information on a particular medicinal plant. We are encouraged to pass that information to the botanical sciences so that they can collect any information that they may have on medicinal plants. We also engage the department in the harvesting of medicinal plants, ensuring that there is sustainable harvesting, because we do not want to lose our medicinal plants. And they are also tasked with ensuring that whenever plant materials are collected, that the plant is properly identified by a qualified botanist, and there is a voucher specimen that has been stored. And they are also engaged with the preparation of the plant materials. But besides that, we also have our department open on every Wednesday, where we receive traditional health practitioners, and sometimes we also receive patients. And when traditional health practitioners come to us, more often than not, they will be seeking registration. Because when they are doing their registration with, I think they do it with the Department of Culture and Social Services, there is a requirement that before they get their certificate of recognition, they have their herbal remedies tested. But we are not able to test because it's an expensive affair. What we do is that when you come with your herbal remedy, we ask them to give us at least 3 medicinal plants that they use in making their herbal remedy, and we assist them with doing a literature search on any information. And we're able to compile a report which they take to the Department of Culture and Social Services, and it facilitates them in their getting a certificate of recognition. Sometimes we also get patients, sometimes they think that probably we treat at KEMRI, we are not able to treat, but we listen to them and we are able to offer them any information or refer them to some of the herbalists that we work with. We do refer to them, or we advise them to see a clinician, depending on whatever ailment that they do. Then we have a chemical sciences division. It's a chemistry division, as the name sounds. And what do they do? So once the plant materials have been collected, the plant is properly identified, they have been graded and properly prepared, it is the role of the chemical sciences to do the extraction, which we can do either using organic or using aqueous. It is the work of the chemical sciences to do the phytochemical profiling, that is trying to identify the different chemical components that are present. And should they need any work, depending on the scientist's need, there are those that just want to work on the total extract. There are those that may request you go further and do your fractionation, your isolation, your purification, and even structure elucidation. And the part of structure elucidation, we do this in collaboration with our collaborators, like at the University of Nairobi, because we do not have an NMR machine. If you may need to use any other equipment, we also rely on our collaborators, locally and abroad. But besides the extraction and the structure and what have you, there is also the key thing, number 4, where we also have some of our scientists doing projects that are looking at adulteration of herbal remedies with conventional drugs. And a very recent work that we did, that should be 2023, not 2003, is where we were looking at herbal remedies being sold in Nairobi, claimed to treat diabetes, and we wanted to find out whether they are adulterated with the conventional drugs. And we did this using LCMS, meaning that we had to develop a method to test the 4 drugs. There were 24 powdered herbal drugs, and we were testing 4, we were looking for 4 anti-diabetic drugs. So you need to, the key thing here is you need to develop a method because we wanted to test them simultaneously. And we also needed to ensure that your extraction method is key. And for more details, the paper is given there. You could kindly have a look at it for more information. But what we found out is that about 4 of the herbal remedies were actually adulterated with metformin, which is very, very risky for the patients. Then for the biological sciences, very quickly, I said we test for safety, we test for efficacy, we can test extracts, we can test fractions, we can test pure compounds. And in terms of efficacy, we are able to do in vitro and in vivo, and we can test for anything for as long as a model or a method is available. But at the moment, in terms of in vitro efficacy, we are able to do antiplasmodial and herpes simplex virus. We do cytotoxicity and microbial, and we have recently introduced Mycobacterium tuberculosis. And then for in vivo, we are able to do a mouse model. We have an animal house, and we can do all the tests that are listed down there: anti-fertility, anti-malarial, anti-inflammatory, and antimicrobial studies. In terms of safety, we are also able to do in vitro using different mammalian cell lines. We can also do in vivo using mice and rats. And right now we are doing acute toxicity, and we are also engaged in determining the medium lethal dose. The other thing that you're able to do is to look for microbial contaminants in herbal remedies, like that paper that I have indicated there, where we are looking for the risk of fungi associated with aflatoxin, and fumonisin in medicinal herbal plants in the Kenyan market. It's also a very dependent research where the researchers went out into the field. They were able to collect a number of herbal remedies in the market, about 100 of them, and they determined the fungal contaminants using pharmacopoeia specifications, and they quantified this using an ELISA based technique, and you can see that 31% of the samples met the standards for microbial limits as specified in the pharmacopoeia, meaning the rest failed. That is how much? 69%. They didn't meet the test. And apart from microbial contaminants, we also check for heavy metals. And we're able to do this in collaboration with Kenyatta University. We also do this with the Ministry of, forgetting the name, where we have the minerals. But Kenyatta University is our main collaborator. We also collaborate with pharmaceutical industries, where, like, for example, the paper that is shown there, we worked with a pharmaceutical industry. They had prepared a herbal remedy for cough, herbal remedy, and it was brought to us, and we validated the safety and efficacy. So apart from our scientists doing their independent research, we collaborate with pharmaceutical industries, even when need arises. And last but not least, we have our pharmaceutical sciences department, and we use them in the tail end. If you have worked on your herbal remedy and you want it now developed into a product, we can engage the pharmaceutical department in the formulation studies. And out of our work over many years, I have highlighted 3 products that we've been able to come up with. We have a nutraceutical in the form of a salt. It's a low sodium salt. We have a Zedupex for management of HSV. And most recently, we have the Tugicide, which is, it's more of for environmental control, to control the jiga free that causes the tungiasis. And this will be my last slide. It's on challenges. Because what challenges do we experience while doing work on traditional medicine? The key challenge is funding. Because you find that there are not so many funding agencies that are willing to fund work on traditional medicine or on herbal remedies, because it is referred to as high risk. And therefore we have to keep on relying on non-traditional sources of funding. And the non-traditional sources of funding, like in this case, is the government of Kenya. And it's difficult. The other thing is in infrastructure. We have to rely on our collaborators for a number of key infrastructures. The other thing is raw materials. Because of the unsustainable harvesting, some plants are slow growing plants. You find that, like for example, the low sodium salt, we've been unable to put it in the market because we cannot get enough raw materials. Where we once relied on, we found that the area is contaminated with a lot of heavy metals. We can no longer go back there for the raw materials. The other thing is a lot of research that we do, it is independent research where we rely on information documented in the literature, in books and in the papers. But we would probably prefer to work with the traditional healers. But it's very difficult to get a traditional healer who would be willing to disclose their herbal remedies recipes for you to work on. And the reason being what Dr. Lydia has highlighted, the issues of IPR. The other thing is that another challenge is the lost traditional knowledge. We know traditional knowledge was passed from one person to the other in particular families. And a lot of these knowledge with time has been lost. We do not have any documented traditional information. A lot of it has been lost along the way. And last but not least is that we do not have a traditional medicine policy. Efforts have been there. I think for the longest time that have been Cambridge. I've heard people saying that we are going to the Ministry of Health, they are working on traditional medicine policy, but for whatever reason, the policy has not seen the light of day. Recently, we were doing a Cambridge bill, and we included the issue of traditional medicine in the Cambridge bill. We hope that something good will come out of it. So thank you. Asanteni sana.
So that is our Kenya Medical Research Institute, meaning that Kenya has accepted that herbal medicines work. And you saw her challenges. It's because she chose to work with Kenyatta University and not the oldest university in Kenya. That's why she has the challenges. And getting herbalists and found them, so look for us. We have solved all those challenges. Any questions for her? Yes.
Thank you so much, Dr. for that insightful presentation. Two quick ones. One, we have seen a lot of Chinese herbal medication in Kenya. Kemri, what's your role in this medication? Because some of them have been reported to be adulterated and all those issues. So I really wish to hear your say on that. Secondly, it has been a challenge to people who want to register their products with the Pharmacy and Poisons Board, because one of the requirements is that you have to declare phytochemicals of therapeutic value, which is so difficult for most of the herbalists. Kindly comment on the same also. Thank you.
Thank you. On the first one, the role of Cambridge in Chinese medicine, we have no role. Because our work, I say the mandate of Cambridge is to carry out research in human health. So when most of those Chinese come and they come with their products, I think they go through the Kenya Bureau of Standards. But in the Cambridge bill that we prepared right now, it's in the Parliament. We've captured some of those issues that we want to be looking at herbal products or any other traditional product meant for treatment coming into the country. In terms of registering your product, if you are registering a product with Pharmacy and Poisons Board, then it has to go through to them, to them you need to declare. They say that probably you even need to have some data on clinical trials, depending on, are you registering it as a drug or a nutraceutical or a supplement. So it depends with you're taking your product there as a what, it is in which category, because there are requirements for a particular category. And I think Professor Oruka is here. She's a pharmacist. She can, I don't know whether you have anything additional. Because I see like most traditional, for most people, they go to Kenya Bureau of Standards. And it's they register it as a supplement or a nutraceutical. But if you go to the Pharmacy and Poisons Board, then the registration requirements are stricter.
All right, just one more now. 1, 2, do I use 1, 2, 3, 4, 5, 6, 7, 8?
So, my name is David Wata. My question is, since you have said that you only validate certain product based on literature, so is it possible for us herbalists to be helped? Because most of the plants, most of them have been documented. Is it possible you people to help us so that we can at least develop a product either in form of food supplement instead of a drug?
I think we have in the past, Cambridge has in the past tried to work with the traditional health practitioners, but it has not worked. I hear they were accused of, the IPR issues are always an issue. it's not easy. We have tried in the past, it has not worked. IPR is a big issue, and because of the IPR issues, the traditional practitioners do not want to disclose their recipes, which becomes very difficult for me as a scientist to work on something that I do not know. So I don't know. Maybe when some of these policies are put in place, they will lay ground on how we can work together.
Yeah. Alright, so we are through. So with all these wonders, now we want to thank you very much. Cambridge, look for University of Nairobi, and you will not have any challenge. So our next speaker is Dr. Ehab, to tell us, it will sing nicely to tell us how these chemicals actually work. We have been told the miracles, the walking miracles. We have been told the Cambry and their policies. Now let's hear from Dr. Ehab. And then after that, over to Dr. Chisetu to manage the other bit of it. Now your presentations.
Since we have really taken a big part of our project on the HPTDC, so I found it would be very beneficial if we can have also some hints or comments on how could we use HPTDC in quality control of herbal products. Okay. So, as we know that herbal medicines are not the same like synthetic drugs. Herbal medicine is a mixture of numerous phytochemicals, while synthetic drugs are pure one single, two, three compounds together that are typically produced under very controlled conditions and consistent formulation. They have also to pass several control checks before they are being marketed to the market. Herbal products, even though they are also quality controlled, they don't get the same, they don't receive the same regulation norms that are very strict, like in synthetic chemical drugs. Plants are living organisms that can be significantly influenced by several factors. This can include environmental factors, geographical factors. So if the same plant species has been grown somewhere and we have collected it from where else, we can really find a big difference in the chemical profiles between these two species or these two plants. The problem is the story doesn't end here. Even many factors can influence the chemical quality of the plants before, during, and after the manufacture or the production of these herbal medicines as well. Each country or each region has their own agencies and authorities which are responsible for controlling the quality of herbal medicines. If we look in the United States, we have the FDA. In Europe, we have EFSA, which is European Food Safety Authority. Even in each country, they have their own FDA. So we can find in Korea, we have Korean FDA. Saudi Arabia, we have Saudi FDA. But they have their own tools to control the quality of the herbal medicines. Whatever the tools they use, we will find that proper identification and testing the quality are at the heart of all of these tools. The first thing to do is to check if you are really using the proper plant species. Have you identified it properly or not? Have you tested if it's adulterated or substituted by other related plant species or not? Companies are required to fulfill or to conform to the pharmacopoeia monographs. Monograph is an official document that can tell the people how to test for identification and also assay or quantitative analysis of the chemicals or even the herbal product as well. To claim pharmacopoeia compliance on an herbal drug, companies need to perform a suite of tests. There are a general physiochemical and microbiological tests they have to follow. You see here on the right, people who are actually familiar with the pharmacopoeia monographs, they can really recognize these chemical tests. One of the essential tests that the pharmacopoeia always use is the HBT-EDC that we have worked with. Those who are also familiar with the monographs, they found that the first step of identification is to use the thin layer chromatography or its modern version of high performance cellular chromatography for identification of the plant under investigation. But what is HBT-EDC? This is what we have already learned together in that course. It's a very simple technique for separation of chemical entities, chemical compounds inside a herbal mixture or even organic mixtures. It started with application, development, characterization, detection, and eventually evaluation. And usually the outcome is in the form of image, which we are always calling HBT-EDC fingerprint. In each pharmacopoeia, you will find HBT-EDC is the first source of identification, the first step of identification, and then later on comes other techniques like HPLC or GC for the assay or quantitative analysis of the chemical markers inside the plant. During the last 7 decades, the HBT-EDC techniques have been also evolved in the authority or in the pharmacopoeia. Until we reach in 2015, we have a general chapter in the United States Pharmacopoeia, and two years later, in 2017, we have found a general chapter for standard method of HBT-EDC in the European Pharmacopoeia. Inside these 2 chapters, you can easily find a method that describes and details how to proceed, how to run a standard HBT-EDC method. When they say in the monograph, you have to comply or conform to the HBT-EDC procedure described, they are assuming that you are following the procedure here. So they are describing the equipment and also the condition that the producer or the company has to follow in order to properly identify their herbal products. It's not only the authority, also the companies. They started also to update and advance this in their instruments in order to cope with the requirements of the regulations. So we can find here during the last few decades, there is a complete portfolio or instrument portfolio for running HBT-EDC, and in 2020 we have found the new HBT-EDC Pro, which ensures full automation of the chromatographic process of HBT-EDC. Automations or instrumentations means automation. And for proper identification, this carry or weigh a lot of advantages. First, speed, efficacy, and accuracy of your measurements or your analysis. You also ensure standardization, safety and environment, increased throughput and productivity, especially with those who are working with a huge number of samples every day or every month. They can easily use the instrument to expedite this process while also keeping the accuracy to the level they want. Let me give you some examples how the HPTLC have been used as one of the quality control standards. If we look at that plant, this is Semisfuga racemosa, which is black kush, and it's a medicinal plant that actually used for controlling postmenopausal symptoms. And usually the product in the market, they have to declare that the product contains only Semisfuga racemosa. But actually, it can be also adulterated by other related species. One of them is Semisfuga foetida, which is liver toxic. Using the HPTLC, we have been able to detect adulteration with Semisfuga foetida down to 1%. As we see here, we have already found this compound that's present in Semisfuga foetida and not present in Semisfuga racemosa. Once you have found this one, you can easily detect that your compound, your product is actually adulterated with other toxic or unrelated species. When we have collected some market samples, they were like 20 in number, and we have found that only 2 of them are conform to what they already declared in the product or in the label of the product. And while 18 of them have been substituted or adulterated by several different other related species. If we look at that plant species, Ginkgo biloba, maybe many of us also know that it has also a lot of medicinal activities for enhancing the circulation in the body and so. Here on the left, we can find that we developed a method for separation of the chemicals inside this plant species. It gives us a full fingerprint for the chemical present in this plant. And we have here some markers that we have already also analyzed in parallel to this plant species. All the way to the right, you can find some of the finished product, which are the product present in the market. Some of them comply. We have found that they are rabies Ginkgo biloba. 3 of them, we found that they are substituents or also adulteration of this Ginkgo biloba. And when the people consume it, you don't know what will happen. This will carry a lot of risks to the health. This plant or this product in track number 12, we found that it is actually Sophora plant, which is a common adulterant of Ginkgo biloba. With that example, we could able to actually identify which market products that are really comply for what they have declared in their product, and also to define what other related species that can be used for adulteration of Ginkgo biloba. That's also another sample of how to use HPTLC to distinguish or identify the Musvelia oleogum resins. This is one of the oleogums that you are using in your research as well, and it's commonly used everywhere in the world because it carries a lot of medicinal activities. But actually, the quality of this Musvelia can be actually altered when it is mixed up with other Musvelia species or even with other oleogum resins because they are morphologically look the same. This method has enabled us to differentiate between Musvelia serrata and Musvelia sacra. It also enabled us to distinguish between this Musvelia species and other oleogum resins, as we see here, Commiphora mer, Commiphora mucor. So we can use it for identification of that one. We at CAMEG have found, yes, it would be very beneficial to have a special method or specific method for each plant species, and that's why we are working on what's so-called the method library. In the website, if you go to the herbal method library, you could able to get access to over 300 methods for 300 plant species that can you freely download it. And once you downloaded it, you could get access to a Word document or PDF document that explains to you how to go through the process until the end and how the identification can be carried out. It also gives you information about atlas. Some images are included inside the mission. So you can compare your results with that atlas and see if you are really having that, the same, if you are getting the same information at this. This year we have adopted a new approach, which is complementary developing system or solvent approach. And this approach simply aiming at harmonizing the development step, harmonizing the chromatographic step. When we look in the pharmacopoeia or even in any other protocols, you can find that each plant has a specific or particular HPT-LC method. And if you are working with 20, 30, 40 plant species, you can end up also with 20, 30 different methods, which is in many cases material, consumes a lot of materials, and also probably also labor, consumes a lot of labor work. But in our approach here, we have used a general method for development. This approach based on creating or establishing 3 different mobile phases. They have a range of polarity starting from low polar, medium polar, and ending with high polar. And we assuming in that case that we could be able to separate the different compound with different polarity using this 3 solvent approach. We have verified it using several herbal products at Quebec, but also we have confirmed it here with the plant species that you guys have used in this research project. This is a paper based on which this approach has been already established. As we have seen here, they said we, in order to simplify and harmonize the routine analysis, so those who are working with unknown targeted analysis, untargeted analysis, they still able to start with that approach instead of going into a specific, because now we are expanding our information about the full picture or chemical constituent inside the plant by stretching the polarity from low polar until the high polar. In this paper, they have used these 3 different mobile phases to read SMS tape. I think this is the one that we have used also in our project, and then medium polar and then high polar. If you put this information on the top of each other, you will end up with a full profiling of what is actually inside your plant material. So those who are working with a non-sample or untargeted metabolomics work, they still able to use this one as a general method for HPT-LC analysis with their extracts. The images that we have collected can be used also, or can be useful beyond just identification or having just nice images. It can be used for quantitative analysis. And this feature we have also applied in this research that year, which is to convert the image into peaks or profile. If we take that track, for example, it's composed of several zones. Each zone has its own color, and this color is composed, of course, of RGB. There is a specific features now in many software, one of them is Vision Cats, that you can convert this color into peaks. And under each peak, you will find the intensity or the area of that peak, so you can use that one for quantitative analysis as well. The peak that you will obtain, or the chromatogram that you will obtain, is corresponding to the number and the intensity to the zones that you have in your fingerprint. One more reliable and accurate way, but we didn't test that in that course because we don't have the scanner, is to use the HPT-LC plate and then scan that plate as a specific wavelength, which is known as the maximum absorbance for the compound of interest. And then you also end up with a chromatogram of several peaks. Under this peak, you have the area, which can be used for also quantitative analysis. Now you can use the HPT-LC for qualitative, identification of the plant material, and also quantitative analysis to assay or quantify the active markers inside your plant. Using these 3 techniques, image results, and also peak profile from image, also peak profile from scanning densitometer, you will have comprehensive HPT-LC fingerprints for the plant of interest. At the end, I would say that HPT-LC is gaining now acceptance in pharmacopoeia. We said in 2015, 2016 is now included as a general chapter for analysis of herbal products. Now we have a concept of comprehensive HPT-LC fingerprints by collecting the images and also having the profile from images or profile from scanning. Quality control herbal products can be now easily done by HPT-LC because we have seen that the first step in quality control is to properly identify your plant species. The future is for automation, and this is what we are working on, and also harmonizing the method for development, and this is what we already adopted in this research using the CBS or complementary development system. Thank you so much for your time, and I wish to be with you next year again.
Any questions for Dr.
Yes, please.
Yeah, thank you, Doc. I just wanted to ask on the use of SST on HPLC because I know of the SST on HPLC, but on HPLC I don't understand.
SST is an integral part of any analytical process. We have actually also SST in HPLC, and we have also created an approach or established an approach for having a general SST as well. This SST solution, which we call Universal HPLC Matrix, you can easily get access to that solution, ready-made solution in the Sigma or Merck. This is composed of 8 different chemicals that you can use to control your chromatographic system. What you have to do is just to run this in one track in your plate and then record the zones that you will find. And you say, okay, this will be my SST solution. So when I run the MPDS, I can find, for example, 4 zones from that UHM mixture. And then when you need to transfer that method from lab to lab or from country to country, then you can tell them the acceptance criteria for that SST. So some pharmacopoeia monographs use the chemical markers inside that plant. So for example, if you are working with a plant who has a specific flavonoid, for example, quercetin, and you have used this condition, you will find that quercetin will be eluted or migrated at a specific location, which we always refer to this as RF. So when you transfer the method to me, you have to tell me what is the SST. So you will tell me, you will find quercetin or whatever, the flavonoid or the chemical marker at that specific RF using this actual conditions. If I can't get the same results as you, then my chromatographic system is not matching your chromatographic system. You have to figure out what is missing and then correct for that to have actually the same SST as you. Yeah? Yes? Polarity, yeah.
So what exactly is the use? Can you go back to the slide?
Yeah, for
the presentation.
yes, that one. Yes, this is based because the compound inside any mixture or even herbal mixture, it is separated different on their polarity. Because we have a silica gel as a stationary phase and also we have a modified phase. And based on the affinity between these two phases, they are being eluted or separated or even retained inside on the stationary phase. One of the factors is the polarity. The more polar will elute the compounds. So we assume that inside our mixture we have numerous compounds with several polarity or varying polarity, ranging from low polar until high polar. And that's why we have created or established this mobile phase. If you look at the polarity index here in this table, you will find that low polar mobile phase having a polarity of 2.6 and high polar having 4.9 or even 5, which are really suitable for high polar compounds like amino acids, sugars. So specific methods can give you just information on specific markers, but general method can give you information about all what you have in your mixture. And this is the benefits of using the CDS.
Thank you.
And Cambry, as you see, this is the best instrument that you can have for it. We already have one at the
Yes, we have now a system now.
I told you all those challenges will go. She needs a microphone.
You can use that one.
Maybe you should give us that equipment, Prof.
No, you buy.
I'm told you has collected dust for a whole year. Can you make proper use of it anyway on a lighter note? I'm just wondering why I'm seeing toluene and a number of labs are no longer using toluene as a solvent.
Still usable, but we are getting rid of petroleum, ether, and hexane. These are the most concerning solvents. Toluene is still even better than them. Actually, that concept is coming from the African herbal pharmacopoeia at the beginning. They were using benzene and even chloroform, I think, and we have replaced that with more safer solvent. And that concept also we have worked since 1 year or 2 years with the African herbal pharmacopoeia. They sent to us a lot of the chemicals and we supported them really too much in order to include the HPT-LDC atlas in their African herbal pharmacopoeia. So you can expect in the next edition that you have also HPT-LDC, and this is what many pharmacopoeias are now starting to follow, is to not only just describe the method or describe what you will see, just also give also some images so you can imagine how your results should look like at the end. Yeah.
February will join us next year. February.
Cambridge,
You know, she's government, so when I catch her, then things go well. Just one more question.
Yes.
For you.
My question is, suppose I want to bring my samples to you.
Yes.
Should I bring them in liquid form or in powder form? And if it's in powder form, should I bring them when they are still wet or dry for analysis? Then I would like to know how much it would cost for somebody to at least obtain a fingerprint of what will be of the end product of.
The thing is that the company is not a service lab, so this doesn't mean that you can send me sample to analyze. Because we are not working as a service lab. We just providing you with the tools that you can help you to analyze. Such project, scientific projects, we always set up with a bigger organization to help them improve their method of analysis. So sending samples to Kamag I think this is not the way that it always work, but we still able to support you scientifically. I mean, if you have a plant species and you don't know how to proceed with that, we still able to help you, send you some methods that we have done before, send you some documents that can help you run the sample by yourself. So.
And remember, I told you we have the equipment. So bring the samples to
Nairobi, the University of Hill. This is a research.
Yes, when you see me, you see him. So bring them.
Is that free?
No.
And the sample is going to be analyzed free.
Did you hear Toluin, all those reagents?
It's now available.
So yeah, at a cost. Yeah, subsidized cost. Yes.
Okay.
Okay. So
Okay.
all right. So
Thank you so much.
and then Kamag also has a local representative. There
Yeah,
she is.
I will introduce
Just-
to you Ms. Claire. Yeah. She is our distributor here and representative in Kenya. She will leading the work, and any support from us can be gone through can go through Claire. I think.
Yeah, so you can get contacts from her over lunch.
Yeah, just
So when you don't want to see me, you can see her.
so just a question. Yeah, I know these machines are expensive, so and a few organizations and universities have some of these equipment. So do Kamaga provide like a subsidized price for like universities and research institutions when they are buying some of this equipment? Or what efforts are you trying to do to see that some of the African universities and institutions have this equipment?
I can answer to that question because I'm not the sales manager, and I don't know how they able to manage such things. I mean, if they can get discount over here to say this is, I really can't control that because I'm mainly concerned with the scientific part. But what I know is we really supporting anybody who is willing to work with the technique itself, because we are not gaming company. I mean, the money that comes to us also goes. It's like a charity. You can consider it like that, yeah.
And those are the type of questions when you ask when you are alone. If we go as a group, you don't get it. But if you go alone, I'm sure you'll get it. Just write some email even to him quietly.
But it's not few machines in the world. We have machines everywhere. Whenever you come to the standard method for HPT-LDC, you will find the Kamag. Because Kamag is actually the world leader on that field. And I could say that almost we have no competitors until.
Like during the training, some of those things you saw, I didn't buy them. I talked to him quietly. Yeah, if you talk like this, you don't get anything. All right, so let's have a round of applause to him. Thank you very much. So now over to Dr. Chiseto to lead the next session, a few presentations before we go for lunch.
so much, Professor Sheila. Good afternoon. Actually, you know, this particular session is your session. The whole of today was supposed to be your session, but I don't know what happened. At least, luckily, we are all here. There are a few housekeeping rules that I wish us to keep as we maintain the program. One is presentation is strictly, all of it, presentation and question is strictly 15 minutes. So talking, if you can do it below 10, that would be wonderful, okay? Number two is I'll wish to a volunteer to take, to be our timekeeper, any volunteer. Okay, that's fine. I hope you're not presenting the first group. No. So immediately 10 minutes are over, raise anything, maybe a black something that you won, and then after your time is up, we'll pick at most 3 questions, then we have the whole day to interact and share. Please take criticism positively from other groups because it's a learning process. You'll go and present this back in your institution, in your conferences. This is like, take it as a big stage. You know those conferences where you go, you're finding 1,000 delegates sitting and you need to talk to them, okay? Take it to that level. And the more you present, the more it becomes easy and easy to us. So at this juncture, I wish to welcome the first group, Boswela Neglectica. Sorry? I'm told there's some. Listen, just a minute. Okay, so we go on. That's fine. Also, that's fine. Yeah.
Just a moment, please. Hello. I guess that we'll be starting everyone off for the IRES final presentations. My name is Tony Johnson.
I'm Eden Yahweh.
And we'll be presenting on Boswellia neglecta. So without further ado, let's get started. We're running through the presentation. We'll do a little bit of background, the study objective, as well as material and methods, the results, conclusions, and future directions, and opinions on where this research could go in the future. So, getting into Boswellia as a species, it's a genus of trees in the family Burseraceae, and it's known for its resin called frankincense. They're moderate to large-sized flowering trees, native to the dry mountainous regions of the Middle East, Asia, and Africa. 4 of the species are considered the true frankincense-producing trees since ancient times, and that's Boswellia sacra, Boswellia carteri, Boswellia frurania, and Boswellia papyrifera. The spiritual, cultural, and medicinal uses of this plant made it so that their value was comparable with gold in the past. This is just a nice map showing the distribution. Like I said, native to the dry mountainous regions of the Middle East, Asia, and Africa. It's adapted to shallow, rocky, and quick draining soil, and it's found in the Acacia conifera bushland ecosystem. Getting into medicinal properties and its commercial use and the relevant species. Medicinally, it's an anti-rheumatic, an antioxidant, an antimicrobial, an anti-inflammatory, and an cancer agent. It's made into essential oils and incense and was utilized in religious rites of ancient Egypt and part of the Jewish sanctuary. A tea is also often made from the bark, and it was also used for tanning animal hides. The relevant commercial species are B. sacra, B. carteri, Papyrifera serrata, and Furania. The ones endemic to Kenya are Boswellia neglecta, which we'll be talking about today, and that was our experimentation, Boswellia revei, Papyrifera, and Ogadensis, as well as Microphylla. These are two images of Boswellia neglecta, one from the University of Nairobi Herbarium and the other from Kew Gardens. It's a less studied species in this genus of Boswellia. They're moderate to the large-sized flowering trees, and it's believed to be more of a hybrid tree rather than a true frankincense.
Yes, so as Dr. had mentioned, there's a lot of commercial use of many of these plants, and with commercialization comes the need for quality control. And quality control always starts with fingerprinting analysis, metabolomics studies, and that is the objective of this study. We conducted various assays to evaluate the metabolomic data for bioactive secondary metabolites present in the bark and leaf samples of Boswellia neglecta. We compared this data to current and past literature in relation to relative abundance and presence of bioactive compounds. This data, of course, will assist in future chemical confirmation of the species and commercial products, and make it safer to use this plant in the future. Of course, the samples had to be collected first. We got them from Mount Kasigao in Taita Taveta County, where they were dried in the ground. And of course, the analytical methods we used were high-performance liquid chromatography, high-performance thin-layer chromatography, which Dr. very well described earlier, and gas chromatography. But first, of course, we have to extract the solutions, to extract the bioactive compounds from the plant material. And we use two different solvents, dichloromethane and methanol. And we should get this done quickly because we only have 15 minutes. Yes, they were sonicated. Sonication allows for very quick extraction, and then, of course, centrifugation to separate out the extracts from the solid plant material. Yes, of course, most of this is automated, so all you really have to do is load the machines and set your parameters, and then the machine will do the rest. Then afterwards, we'll come in to interpret what the data that the machine generates. What is more hands-on at the moment is the HPTLC, where we have several relatively manual processes, but a lot of the machinery does semi-automated to make it easier and much more precise. There's the application onto the plates, onto the stationary phase. There's the development using the aforementioned solvents, the low polarity, medium polarity, and high polarity solvents. Then derivatization to make various compounds, once separated, more visible, so we can actually image them properly with a visualizer or a scanner. Most of these are provided by Kamal, manufactured by Kamal. Then, of course, the data analysis is done in their software Visual Arts.
Getting in the first of our results, we're looking at our liquid chromatography and mass spectrometry data. So this is showing our peaks for Boswellia neglecta, and I'm sorry. As you can see, there's multiple peaks, and they're wider than you would normally see if it was a singular compound. So it's labeled just as sesquiterpene rather than being an individual compound, as there are multiple sesquiterpenes or compounds that are found in Boswellia as a genus that might be available, but there's limited literature on Boswellia neglecta, so we are unsure if it is actually a specific compound. So being that there are multiple that it could be, and that there are multiple peaks in the same ion zone and in the same molecular weight as a lot of different sesquiterpenes, they are labeled differently, but in the bark you can see we have the 3 peaks, and the leaves, the 1 large peak. This is continuing on with that. This is showing some of the sesquiterpenes we have from our molecular library that could be possibly available. The species, once again, is understudied and not considered one of the true frankincense. So this is data that we can pull from the other genuses or some of the true frankincenses. So looking at pyrophyllene, gerginine, and humulene, as you can see on there, they have a molecular weight. It's in the same ion zone that we found in our LCMS data, but because we don't have chemical standards available in the lab when we did our experiment, we can't be sure exactly which one is which or exactly which peaks they are. In our GCMS data, we had 66 identified compounds in our leaf samples and 62 in the bark. And this is 2 graphs showing the 14 of the most abundant compounds from each. On the left is our leaf, on the right is our bark sample. You can see that some of them carry over to the other, but there are differences. Alpha pinene is one that matches on both sides. And if you look down, you can see that the abundances are different as well as the amines. This is just another graph showing our GCMS data. It gives the molecular structure of our compounds, as well as the retention times, the area, and the library ID that you'll find is familiar from the previous slide. Looking at our leaf sample and our bark sample, we labeled the highest peaks found in the GCMS chromatograph, just to get an idea of what it exactly looks like and the abundance of each of our samples.
Yes. After we identified a number of compounds from our gas chromatography, we conducted another literature review, a secondary literature review, to find out the various bioactivities of many of these compounds. And we found anti-inflammatory, anti-nociceptive, antimicrobial effects, which does indeed validate many of the traditional uses of Boswellia, and even Boswellia neglecta, which is used as teas, tonics, and of course as poultices, which has traditionally been used to treat arthritis. Notably, of many of these compounds, at least four on this list have antioxidant properties, which will explain the antioxidant assay which we performed during the HPTLC, which I will detail here. Yes, during the HPTLC, we separated one plate to perform an antioxidant assay, in which we replaced the derivatization reagent with DPPH, diphenyl picryl hydrazine, which allows which gives a demonstration of how much of the antioxidant properties of the various biochemicals that have been separated out. And as you can see here, on the left is the blank. You can see the white yellowish area. That is a display of the antioxidant properties of the Boswellia extracts. Yes, and here are the general, these are our general results from the rest of the HPTLC. Top to bottom, we have the various solvents, low polarity, medium polarity, and high polarity. Going across is the visualization under different light conditions. We have shortwave UV, longwave UV, and white light. Yes, as you can see, the first one, the low polarity solvent, gave us the best separation. This is important to note because when we're talking about resins, we're talking about terpenoids, which tend to be fairly low polarity, so we want a low polarity solvent to separate them out properly. In conclusion, our investigation found many compounds which allow for the bioactivity of the compounds we found does indeed confirm the traditional uses of medicinal, uses of Boswellia as being valid. And we also found several anti-cancer and anti-genotoxic compounds which can inform future pharmaceutical use and warrant further investigation. There is a noted absence of boswellic acids, which is important because those are some of the key compounds found in Boswellia sacra, Boswellia serrata, and the other true frankincense. And these are the products, these are the species that are used commercially most. And of course, we have to be concerned about adulteration using the more common species that actually don't have some of the more important biochemicals. And of course, most of this is done using GC because, again, terpenoids, most of the relatively small terpenoids will be most recognized in GC. And we didn't find much literature that was studying the much larger compounds.
Getting into our future directions and just finishing up the presentation, Boswellia neglecta is understudied and there's not a lot of literature that prioritizes it, as well as the other species that aren't considered true frankincense, but they still have something to offer to research-wise. Further in-depth investigation and other members of the genus, besides like the Boswellia sacras and serratas, can lead us to more empirical evidence that could lead to more exciting study in the field. So conducting this experiment with more replicates as well as laboratory chemical standards of the potential compounds would help us gather more empirical evidence and further elucidate the profile of the understudied species. So thank you, and That was us. Questions, I think.
Who told you to clap? I'm the boss here, okay? Please give them a loud clap. Leg clap. Leg clap. That was really wonderful, okay? I really appreciate that. So right now I'll take 3 quick from the floor and then, wow, first hand is here.
And very, very wonderful presentation. Thank you so much. I was interested in I wanted to know whether Lydia and her group can relate to this. Is this any plant that any of you know about? Number 2, I was interested in wanting to understand, because I saw you looked at the bark and the leaves in terms of the compound distribution. Was it different? I know that the Lydia's group, some of them prefer the bark and sometimes the leaf. In your group, in your studies, was there a difference? Should they just use the leaves anyway? They're easier. When you go cutting bark, that's what I was doing the whole of last week. And they would not allow me to cut bark. They're like, no, but if I'm taking the leaves, they were just fine.
So with respect to that, I'll answer that with a bit of anatomy. The resin ducts, which is what produces the resin, or what actually secretes the frankincense resin, that is what most people are after when they talk about frankincense. And those are ubiquitous throughout the plant. You can even get it out of the roots, but damaging the roots is very risky. So you do get a lot of the very similar compounds throughout the plant. There are more resin ducts in the bark. So if you want more of your terpenoids, that's where you should probably go. But harvesting resin from the bark is also very resource intensive. I think the leaves are usually the safer option as far as preserving the life of the plant. As for compounds, there were differences, but not very much. And of course, this would require further study because that is another thing that varies significantly based on the growth conditions of the plant.
But your own plant that you studied,
Yes.
you look at the leaves, you look at the bark. My question is, how did the leaf and the bark compare in your studies?
Yeah, very
In terms
similar.
of,
Yes, here the most abundant compound of the leaves that we had reasonable certainty of was layered in the bark, and the leaf was pyrrolidine, which currently we couldn't find any report of activity. But the next one is amarin, which is a pentacyclic triterpene. So is oleanine. So these pentacyclic triterpenes, which are similar to the boswellic acids I mentioned before, they're also very important. They do have a lot of this very similar bioactivity. So likely you won't find very different uses between the leaf and the bark, but maybe some differences in optimization. Again, further investigation, further practice.
Maybe just to assist you answer that is, you could have done principal component analysis whereby you take the data for leaf, take the data for bark and do the statistics, try and see are they different, and then be able to conclude. But the way it is, you can't say much about your data. Okay? So time is oh, Lydia needs to comment something.
Hello. As for the traditional healers, okay, we have been using the root and the bark, but of late, because of the conservation and the consistent supply of our medicines, we have opted to use the leaf. And I think most of the traditional healers, they know that plant, they have used it from time immemorial, and most of us who have started conserving the plant, we are emphasizing, even for the researchers who feast us, we should concentrate on finding out which part of the plant works best. If the leaf, then we will be on the winning side. Because with climate change, global warming, as much as we'll say we are going to domesticate the plants, there's that effect. How much supply are we going to have? So I think our recommendations of late for the plants which are rare species, we're concentrating on leaves. like Wabuge Ugadesis and the rest.
Thank you. I'll give Dr. Peng just 1 minute and then I'll wrap up that session.
Since when you answered Dr. Osano's question, you said if you're looking for terpenoid, you go to the bark. Since the bark probably will produce the resin, probably have more terpenoid. So, but basically, you want to answer the question, use your data. So if you see your GC data, in fact, the leaves have more terpenoid. If you go back a slide, you have the chromatogram and structure, you see, your leaves have more of this terpenoid than your plants. So that's also raises a question. Since the resin is from the bark, why the leaf have more resin than the bark? So that's the formula to think.
Yeah, think about the metabolomic pathway. Where does it start from? Does it start from the leaf? Does it start from the bark, and so on? So I recorded some few points of improvement. One was one of your slides for introduction was too busy. Rule of thumb: when you're giving talk, please have a maximum of 3 lines per whatever you want to talk, not a whole paragraph, right? You gave the location of the plant. Please also go ahead and give out the GPS points, because if you say Mount Kisaju, Prof. Osano doesn't know where Kisaju is. Okay, she went there, but somebody else in the room, they don't know where it is. But if you give a GPS location, it will be like universal, the way you call Exen. If you say Exen, somebody in the US will know what Exen is. So if you give a GPS coordinate, that will be so wonderful. And then, this is a general audience. Okay, unfortunately, we are all similar, doing the same thing here, but when you go to a bigger platform, don't say shorter wavelength. You know, it doesn't mean anything. Say maybe when you look at the UV spectrum or light spectrum, you need to specify what was the short wavelength, what was the long wavelength or medium wavelength that you're looking for. Not just because it starts from 190 nanometer all the way to 900 nanometer. So be specific on what you're working on. But overall, you did a good job. In terms of time,
We're over.
no, not the talking, their presentation.
Oh, yeah, they were like, I think, 30 or 60 seconds. Whatever they did well.
Okay, that's fine. We give them a 90%. Thank you so much. Okay, so I welcome the next group, Erythenia, Abyssinica project. So please come on.
Hi guys, how's it going? Feeling a little nervous, and I want to say, Dr. DeSetto, this doesn't count towards my 10 minutes. I'll still try to talk pretty fast. Well, this, I guess 2024 now, I will be presenting on Erythenia abyssinica with my partner Dan, and of course, my other valuable partner, Sumala, that unfortunately cannot be with us, but she's here in spirit, right guys? Yeah. All right, well, we can't really start talking about this program before we talk about traditional medicine, right? So, the traditional medicine is the application of these medicinal plants to treat ailments and illnesses. But the important thing is, research, and I mean Western research, is finally starting to accept this ancient knowledge as a substantial contributor to medicine and health. And this is all derived from plant secondary metabolites, and that is what our whole research project is about, right? Classifying these secondary metabolites through cutting-edge analytical techniques. Dan, I will let you tell us about our plant.
Thank you very much. I hope you're all doing well. My name is Dan. So Erythenia abyssinica is our plant that we deal with. You can see it there. It has a lot of secondary metabolites, and when I say secondary metabolites, you need to differentiate from primary metabolites. Primary metabolites normally are those which plants need for their growth and survival, but secondary metabolites, they play other ecological roles, e.g. being antioxidants. And for this plant, it has a lot of secondary metabolites, be they flavonoids, they are alkaloids, we have terpenoids, phenols, and quinones, plus many others, okay, including coumarins. All right, and we can see here that some of the secondary metabolites they have play so many pharmacological roles. Okay, most of the literature claim that they are anti-inflammatory, they are antioxidants. They are also used for management of cancer, diabetes, They have antibacterial, antiparasitic, and antimicrobacterial activity. And a good example here, for example, erisodin here has curare-like activity. Curare-like, we mean it blocks neuromuscular junction, you know. NFJ is where the neurons meet with the muscles, you know, and therefore it means it can cause paralysis of muscles, just like curare and tubercularin, which is a drug that has been developed from there. We also have chalcone, which has antioxidant activity, and the antioxidant activity, you can check based on the structure that the many double bonds it has can actually harvest photons, that's light, and therefore prevent oxidation. All right. Why the study? Okay. Because of the claims of pharmacological activities of the secondary metabolites, we had to find out whether this is true or not. You know, and by using HPLC-MS and GC-MS, we went out and analyzed the secondary metabolites that is present in Erythrina abyssinica. Okay. And therefore, we compared with what is claimed in literature plus what we found out. Some of the materials and methods that we used. So the samples, we bought the samples from an area called Muteteni in Mola Sub-County in Machakos County. But I think as Dr. Tisito has said, we should have included here the GPS points for our location. And we analyzed this using HPLC-MS and GC-MS in Isipe and HPTLC in Chiromo. Let me allow Ashton to proceed with the methodology.
And obviously, we've all done it, so we're not going to take too much time. But one thing I really wanted to focus on is if we're looking here, this is the entire extraction protocol for HPLC, which was also used for HPTLC. If we look at the equipment, very basic laboratory equipment. We had a sonicator, we had a centrifuge, and we used some methanol and a paper towel filter, right? That'll always be a memory. So whenever I work with plants, we always say they're nice because at the basal level, they're simple, but then you can always take it on up to just apply some of the most amazing technology I've personally ever seen. Now the parameters is something you can look at in our review, so we'll go through those. And again, as I said, with the HPLC, GC is exactly the same with the exception of we are using a different solvent for the volatile compounds and a little bit of drying agent so the water doesn't really mess us up. Now, HPTLC is really the shining star of this program, right? How many of us have used an HPTLC machine before here or even seen one? All right. Well, back in the day, me neither. And again, we're seeing a relatively simple process with 1, 2, 3, 4 steps, and we get these beautiful, beautiful chromatograms that can be used for very, very quick analysis. Now, our system here is not the HPTLC Pro, unfortunately. I know Dr. Ahab would like to get us in touch with the sales department, but resources. But we use a semi-automated process where a Linomat played at 0.2 microliters to 0.6, depending on what our group was. And that allowed for what? Replication, right? Anyone with our parameters should be able to obtain the same results. Furthermore, we use a complementary solvent system with low polar, medium polar, and high polar. And then we took images at several different lights, including white light, so what we're seeing with our eyes, but then also UV, because as we look at the chemical structures, we're seeing a lot of benzene rings, we're seeing a lot of these compounds, and most of them fluoresce under UV light. So that's very important for identification. Now, we went through what we did, but why did we come here? For my U.S. fellows, I like to assume it was for scholarly advancement and not just a free trip to Africa. Maybe kind of. But this allows us to classify the secondary metabolites that we're seeing in traditional medicine, i.e. the intersection of science and tradition, which is such an important thing. I know Dr. Osano loves to talk about the religion and science, but tradition, science, we're all in a web, right? The web of humanity. Now, before we go any further, we have to take a quick note on the medicinal uses of erythrina. Now, they're well characterized, but almost every part of the plant is used. You guys probably can't see that because I can't, and I'm much closer. But you basically take extracts from the root, the bark, the leaves. All of them are used in traditional medicine, but with different applications. So by looking at the extracts from the different plant tissues, we can see, hey, do we agree with what is traditionally used, or can we make it better or more sustainable? Because in this era, sustainability will be key. That's why we like Western medicine, right? We have a chemical reaction, we send it to the manufacturers, they do it by the ton. Now, what did our results look like? Well, you guys are actually lucky. I remember the first year I did this, we didn't actually have the pleasure of doing HPTLC. We got it directly from ChemEgg, and it was like a 200-page PDF. I almost had a heart attack. But with any Metabolomics study, there's a lot of data, and we have to analyze it. Now, we used HPTLC with visible light, and here we see beautiful, beautiful chromatograms. But if we take a closer look, what are they telling us? Well, this is the same solvent, this is the same solvent, and this is the same solvent. So we're actually seeing distinct differences between the different tissue types. And if we look under UV, we see the same story. And we also can see maybe leaves are kind of hard to quantify because of all the chlorophyll backgrounds. Now, another nice thing about HPTLC is you can directly apply bioassays. And it was a DPPI antioxidant. And what we found was erythrina, in kind of like how it says in the literature, we have some, well, let's just say discernible antioxidant activity. We really should have probably done a 15-minute, a 20-minute, and a 30-minute, but we're seeing something, which then corresponds to the UV light to show, okay, we might have some bioactives. Obviously, this is something that can be optimized. But the limit of HPTLC without our standards is we really don't know what is there. So we moved on to LCMS. Now, back in 2021, a very, very thorough review paper of erythrina was published with almost all the known bioactives. So what we did from here is we created our molecular libraries, and you do it either n+1 or n-1, depending on mode. We wanted to say, hey, is what they claim actually in our sample? And we actually did identify about 3 compounds in each mode that corresponded to the literature. GCMS, however, provided a more novel approach, where we are seeing that, hey, we had 35 to 37 different compounds that were not appearing in literature, some with medicinal applications, such as hexonic acid, derivatives, stigmerstol, and squalene. Now, relative abundance was actually very similar between samples, and I know I have about 45 seconds, so I want to pick it up a little bit so my partner can do a little bit of talking. You guys are probably sick of me by now. But despite their similarity in profiles, one thing to say is this right here is actually a major bioactive of the hexonic acids, which are antimicrofungal and a little bit antioxidant. So I'll let my partner do end result. You got 30 seconds.
Thank you.
Dad?
All right, so in conclusion, what did we find out? That most of the compounds that we found out were similar in previous samples, but they were not available in literature. Okay? And that a large number of those compounds that were identified in literature were not found in our sample. Okay? And furthermore, that optimization is needed for the techniques that we used. That was our conclusion, and we thank you very much. Any questions?
We've got 4 seconds.
Yeah, that's fine. Give them a snap clap. That was really nice, and I really appreciate their talking, whatever they presented about the erythrina. I'll take 3 questions from the floor, and then we have a wrap up for this session before the next month. First question is here.
I think mine is not a question, it's a comment on the way you are writing the erythrina abyssinica. I'm not a botanist, but I'm told the name of the genus should be capitalized and the species should start with a small letter. You wrote, yes, so you used capital for both of them. And then there's the slide just before the methods.
Slide just before the method. This one? No.
Just before. Yeah, that one. The second statement that they are currently no. I was wondering, therefore, identifying secondary metabolites through these techniques. Did you do your literature search? Because I know there is a lot of work done on Erythrina abyssinica.
Oh, yeah, so as I said, in 2022 there was a huge article. I think it identified almost 180 different compounds, which is what we use to build a molecular library for the LC. But despite this vast knowledge, when we say there's no pharmacological drugs, we mean like by the Western sense that are FDA approved, this, that. Herbal supplements, yes, but we have not isolated the secondary metabolites and used them in a direct application for pharmaceuticals.
Why not use what is already isolated? What is the why the isolate? Or was there anything new that you are looking?
So with GC, we did discover several compounds that do have well documented in other plant species medicinal values. So the purpose of this program is for quantification. And what is the value of these, or rather potential value, in future pharmacy and of course economics?
So it's well known that you have been to this program before.
It's supposed to be a secret, but what's up?
But you did the same plant then, meaning that this is essentially a replication study. How well did it replicate your results from last time?
So that is a very good question. As I believe I had somewhere in the slide, most of the novel compounds that we found last year are making their appearance again in the GC. But last year, the chromatograms were quite a bit different. I noticed we saw a lot more variety, which is why in the final stage we were saying tissue composition varies a lot. This year it seemed a lot more precise, or rather aligned. So if we actually look at the chromatograms, the HPTLC was very clearly different, but the GC, the amount was almost the same, as well as the compounds that we found. The only difference was that leuconol, which is an anti-cancer agent, was only found in the bark, which we found to be interesting.
Maybe just to add on that is, you did not collect from the same plant you did last year. Even if you did that, it's
So
1 year down the line.
So we did actually.
No, if you did that, it's 1 year down the line, so you don't expect it to be
Exactly.
the same. So there are quite a number of things that maybe I need to raise up. One is, like the first group, they said they didn't find out boswellic acid. You know, we gave out a generic extraction method. So if you are interested in targeting boswellic acid, then you're supposed to look strictly on which solvent can pick up boswellic from the extract and use it up. Okay, it was generic. The same to this other one. You see, like even them, they could not read whatever they're presenting. So it's a point of improvement. Like when you present out, please let people be able to read from far. And don't be too lazy. It's like your first slide, you just cancel 2022 and then add it 2022. Let's be too lazy. Okay? Please try and look into that. I'll give you a chance. And then you gave out the footnotes 6, 8, and so on. It's like you're picking up from literature without having it out. You know, like when you reference something on the text, then just have us a footnote below. Tell us maybe it was Peng et al. Or somebody who did that particular work. Instead of giving numbers, doesn't mean anything.
It's good. The reference slide is
At the end, yeah, it doesn't mean anything. So in the same slide, add a footnote below telling us what that 6 means. So maybe you can just have a small sample, something like 6. What does it mean? This is not important. The last slide is not important, but it should be within the slide so that somebody looking at the information should be able to quickly reference that up. I know time is running up. I'll just allow you 30 seconds to say something.
Thank you. 30 seconds. Ashton, this is your second. Please, what will you do different? This is your third or so. What will you do differently? Given another chance. Thank you.
Well, if I had another chance, as Dr. Seto said, we've used very generic extraction protocols, right? Which are not exactly optimized to the plant. As we saw with the HPTLC, solvents make a huge difference. Can you pull up the HPTLC data? Because I had like 5 seconds now. Okay.
You got 10.
I got the whole 10, wow, but there's no 6 of them. So you look here, solvents, huge difference. That just kind of emphasizes the need for optimization with these. Methanol extraction, as Dr. Seto said, it's generic. We're kind of doing an overview, but for a lot of the targeted compounds, we cannot find them. That's why we did not find a single compound in the GC that was documented in the literature. The LC we did, but we only found 3 out of 120. So.
Okay, that's really nice. So please, people in the afternoon, I don't want to see percentage up to 4 decimal places because that is very huge error. These percentages have it up to 1 decimal place when you're putting up your abundance. I saw in the morning before we are breaking up, people are giving out percentages as 1.004%. That's a very big margin. Strict it down to 1 decimal place. So at this moment, I want us to go and have a break. And as I told you, we have so many reasons why we eat, and nobody has the true or right answer to that, right? So still think through it. Why do we eat? And then we are breaking up. We're coming back at exactly 1:30, and at that 1:30, the moment we waste here, we're eating into our lunch. So I hope every one of you has a lunch voucher, because you won't be allowed to eat without lunch voucher. Yeah, Professor Sheila will give you one. And then exactly after lunch at 1:30, we are here to maybe get an overview of what the Azaredica group will be showing to us. Okay? Any other comment or point before we take off? General point or announcement? If none, then Professor Sheila, where are we having the lunch?
Just at the entrance, the dining hall, just at the entrance.
Okay, enjoy your lunch.
You can't miss it, just follow this.
Yeah, don't eat too much.
Next time,